Okay, Matt. So let me formally introduce you. So Matt is going to present his talk on causal inference for genetics guided drug discovery. He is working at 5 Prime Sciences. And over to you, Matt.
Perfect. Yeah, thank you very much for inviting me to be here. I'm talking from Toronto, so a little bit earlier in the day than the rest of you. And yeah, I wanted to, my talk's gonna be fairly light on kind of technical details, but just give you a kind of overview of how causal inference is applied in the field that I work in, which is yeah, human genetics guided drug discovery. So I'll start with what we're doing now. And then the thing with our field is it's sort of in flux with the amount of new data, both in terms of what we can measure and how many people we can measure it in. So really, the focus is asking the next questions for our field, and I wanted to sort of pitch a few of them to you at the end. But currently, what we're doing now. So yeah, my name's Matt. I'm an associate principal at a company called 5 Prime Sciences. I did my PhD at the University of Bristol in the MRC Integrative Epidemiology Unit, and Qingwan was one of my supervisors who I believe is here now. And 5 Prime, basically, it's a small consultancy spun out of Brent Richards' lab at the University of McGill, and it uses human genetic evidence and multiomics to try to identify better drug targets, identify biomarkers, anticipate side effects of incoming medicines, and help select the right patients for clinical trials. And the reason we use human genetics for drug discovery is very simply that drugs succeed, not always, but often when, or certainly it's a necessary condition that the target has a causal role in the disease. And a recent review paper that was looking at just how valuable human genetic evidence can be, and the limits of that evidence kind of made the claim that human genetics is one of the only forms of scientific evidence that can demonstrate the causal role of genes in human disease. And the reason genes are important in drug development is that the vast majority of medicines are targeting either the product of genes or the transcripts of genes directly. So they have this figure here where they try to show the enrichment for the likelihood of a drug succeeding through phase 1 to phase 3 clinical trials, given that the gene that it's targeting is associated in some way with the disease that's being tested in the trial. And they find sort of between a 2 to 3-fold enrichment, which is not huge in magnitude, but when you have a sort of 90% failure rate, this does kind of help to reduce that a bit. Two sources of evidence I'm not going to do a sort of primer on in the interest of time, but kind of the workhorse methods in this field are what are called genome-wide association studies, and then a post-GWAS method called Mendelian randomization. Just to give you the terminology, a GWAS very, very briefly tests the statistical association between one copy of a particular letter in human DNA versus another, and whether people who carry one letter have a higher likelihood of disease compared to those who carry another letter. So it's a very simple test. It's just a linear regression. And the causal foundation of the reason that we might be able to ascribe a causal interpretation to that association is that letter differences in DNA can, under certain conditions, be thought of as being inherited randomly at birth. So whether I have one copy of a gene versus another, that is sort of or under certain conditions is a random process. So I can view inheriting that copy as causing an increased risk of disease or causing a change in my cholesterol levels or my height or my weight or whatever it might be. So I'll very quickly go through a real example. This was a client that had come to us called Maria. They wanted to test a gene called ANGPTL4 to whether it would be an effective medicine for reducing triglycerides or coronary artery disease. And so when we looked at the evidence inside the gene ANGPTL4, there was a genetic variant called E40K, and that rendered this protein completely non-functional. So people who carried this variant did not have a functional copy of ANGPTL4. And in dual studies, carrying this variant was associated with both lower triglycerides and lower risk of coronary artery disease, which were the two outcomes they were interested in. And so the idea here is that this could mimic the pharmacological effect of therapeutically inhibiting ANGPTL4. I have a large group of humans. Some of them carry no functional copy of this gene, and their triglycerides and coronary artery disease risk are significantly lower. So perhaps if I medically induced that loss of function in ANGPTL4, I would see the same effect. And for this client, one of the major issues was that ANGPTL4 had been tested in mice, non-human primates, rhesus monkeys, and these animals had developed a disease called abdominal lymphadenopathy, which is kind of a swelling of the lymph nodes in the abdomen. But what we found is that in humans with 2 copies of E40K, so they are producing 0 functional ANGPTL4, there is no abdominal lymphadenopathy in humans. And when our client took this to trial, of course, this is very much, this is an anecdote, but this was kind of an exemplar setting where when they went into a phase 2A with their ANGPTL4 inhibitor, they found that it worked very well. There was a 58% reduction in triglycerides, which is exactly predicted by the E40K variant that causes a loss of function in ANGPTL4, and there is no evidence of lymphadenopathy, which is what was predicted by those individuals who carried 2 non-functional copies of ANGPTL4. So lastly, I realize I've only got a few seconds left, so I won't spend a huge amount of time on this, but the issue we're facing now is that we're measuring a lot. So there's an explosion in our ability to measure transcripts of genes in different tissues, metabolite products, proteins. For example, these 2 assays here are able to measure thousands, tens of thousands of proteins in circulation. The UK Biobank Pharmacoproteomics project is going to increase to 500,000 participants who will all have these thousands of proteins measured. We're getting thousands of proteins measured in cerebrospinal fluid, in brain, for 800 metabolites. And this data is sort of growing. The dimension of the data is growing. And it's sort of outpaced our ability to have methods that can easily sift through and generate exposures that are useful. So anyway, I'll went a little bit slower than I thought, but basically the main issue here is that we have too much data and not enough methods to deal with it. Thank you very much.
All right, thank you, Matthew.